My name is Max Nerlansky, and it's my pleasure to introduce Flowcell to you. So Flowcell is a kind of a culmination of my personal dream to turn scientific knowledge into an actual contribution to humanity, specifically in the field of healthcare. I've worked in years for diagnostics, but there is a very big problem that's facing us right now, and that has to do with flawed drug development processes. And this is of course very pertinent in the age of COVID. So the attrition diagram on the left shows the time frame and the attrition of the drugs during development. So this is due to flawed models, especially in the preclinical models. So the technologies are old and inefficient. And though we kind of feel that we're in touch with DNA via CRISPR and other methods, really we have very crude and inefficient ways of doing a lot of pipetting. But more importantly, development is blind. So in the age of COVID, this is particularly problematic because by and large, we're not using individual biomarkers for precision medicine. This is starting to come into vogue, but is not actually happening for COVID, which is a huge problem. But specifically, we have a shortage of time, and even the efficacy of the drugs as they're being developed is jeopardized by the flawed processes. So what does the solution look like for COVID? Well, let's think retroactively. We want to deliver precision medicine. That requires identifying the state of the disease with biological information derived from a person, biomarkers. They could be cells, proteins, etc. What is lacking, as I said, are the tools to do this in clinical settings or to develop quality biomarkers using good representative physiological models. So these tools include techniques like flow cytometry, electrochemistry, and optics, of course, with miniaturization. Here are the applications. Diagnostics, point of care diagnostics is an area I've worked in for a decade, but it has high regulatory burden. So the entry point to the market is actually the research use only areas of research that will enable development of effective biomarkers. This field is growing very rapidly at 20 to 25% year over year, driven by 3D physiological models and organoids. But the clinical market is even larger. And of course, there the needs are huge and the market is in the billions. What do these solutions look like? They are providing this biomarker bridge from preclinical work to clinical to the new drug approval. So the traditional model has this separation between drug development and precision medicine biomarkers. So there's a gap of translational value. Microphysiological systems bridge that gap and feed straight into precision medicine or companion diagnostics, which is how FDA wishes to see drugs developed in the future to be jointly co-developed. So these two regulatory departments are working hand in hand. And the systems can look like this. I'm not showing this one because it'll be revealing too much. This is an actual integrated micro-optical chip that's being licensed. But as I said, the microphysiological systems for research are the entry point. So here's the kind of product development pipeline. The flow cytometry, which can be an element of this, of course, can also be a standalone software. So that already is in place in need of beta testing. There is a distribution aspect to this, as I mentioned, but the first elements are for preclinical. So this is the timeline. The competitive landscape looks like this. There's a lot of white space, and generally speaking, the fluidic control and the high throughput are not satisfied. So this is what Flowcell will address. And we have a team that spans the necessary expertise from microfluidics to biology to business. And here are some domains that explain some of the vision and technology. And please feel free to reach out. Thank you.
Thanks.
Thank
So
you, Max.
any
Perhaps
questions?
I'll go again.
I should mention very briefly, I have left out a number of supplementary slides, for clarity. So, yep.
I'm sure you have. I'm not a life science person, but just understanding the nature of the problem. You said very early on that the process was flawed, that tools were possibly lacking right towards the beginning. Perhaps I could ask why against the problem. Why are these things as they are? How have we got to this place where this process is?
I would say it's economic incentives. So in that marriage of drug development and drug diagnosis, the companion diagnostic that takes a more holistic approach, diagnostics is the Cinderella of the story. So it's not a marriage of equals. There's been growth on the development and diagnostic technologies. And you don't have to go much further than the COVID story to kind of see that. But also with drug development and vaccine development, we can see how this problem of all of the attention and resources of the world just cannot be reduced in development time. So I think the evidence is there. But specifically for cancer, which is my specialty, the statistics are very, very poor. Generally speaking, after years of preclinical development and hundreds of millions of dollars, clinical success rate getting to the new drug approval is only 5 to 7%. So less than 1 in 10 make it after all of that effort. And that's because of flawed systems being baked in the cake. Thanks.
So 2 questions. First is a quick one. What cellular models have you tested already for your system? Second question is, what is the impact on cellular viability, function, differentiation, morphology?
Yes. So I'm primarily going to have to refer to literature on that. In one of these domains that are mentioned here at the end, physiological models, there is a white paper that I've posted there, even kind of official publication, that talks about existing models that utilize organoids and flow. So the short of it is that the most pertinent models, especially for COVID, are lungs and gut, which is where the infection occurs. So there's been a noted improvement in organoid, which is kind of a minimal biological entity, right, to capture that physiological relevance. There's been noted improvement in their viability. And certainly people have talked about, I can point to specific references on the improvement of relevance of biomarkers. So it's mostly, I would say, literature, because these platforms are under development. One of my collaborators has a paper that's in publication right now, so I can connect you to that, but I'm more on the engineering side and partnering with biologists. I hope that answers it.
Not exactly. So basically, my understanding is your platform have already at least tested some of the models and already got some data, right?
No, not my platform. So there are several elements to this. So while there are widgets, as I'm calling them right now, being developed, that is still in the works. But as a immediate value kind of delivery, there is a website that provides some of the off-the-shelf solutions from existing suppliers. They're very hard to find. Some of them are even in beta stages. So I'm popularizing that. So that's some of the data that I'm referring to. This is extremely early, kind of development technology.
Okay, thank
sorry,
you.
This is Robin from Wyk.
Okay, very quickly.
I just want to understand exactly what service would you be providing, because ultimately in drug discovery, a scientist goes through target validation and identification. Well, first identification and then validation, and then goes through high throughput screening to find a compound or a molecule that can modulate the target. So obviously, I totally agree that having a perfect biomarker is a wonderful thing, but it's just not always possible and available. So ultimately, how do you plug yourself into the world of drug development? To who do you provide your services?
One of the well, there's several segments. So there are certainly scientists in pharmaceutical companies. There are researchers, but maybe one of the most logical segments is one that you've alluded to: high throughput screening. There are CROs that do a lot of that and specialize in it. So the very specific value add that I'm providing now via distribution, before some of the things from Flowcell are ready, is a transition from 2D to 3D screening, and also to use of organoids. There was a conference with that exact name in Boston in March that has been postponed to July and now to August. But this is very much a relevant biological model to go away from 2D inadequate model in which you can develop biomarkers, but they may not translate as well. So by having these improved platforms for developing biomarkers, you can do better screening. And it is somewhat of a question how much 3D is necessary for high throughput screening. Some of the customers I've talked to say that they like to do medium or low throughput screening in high value physiological models, and then for truly high throughput screening, go back to simplistic models while keeping the biomarker. If that logic makes sense. Develop the biomarker in a more complex model, and then for high throughput, go back to the simpler, cheaper one. But certainly there's a lot of need for this relevance before the drugs go into humans. The pipeline shows this.
Thanks, Max.